Faiza, are other students also coming or not? Am I audible? Anas?
Yes, ma'am.
Am I audible?
Yes, ma'am.
If you go through the models I posted in the previous class, actually there was no net, so we couldn't go through those models. So I'll revise them in today's class only. Okay. Shall we wait or start? Okay, so I'm sharing the screen and we'll start discussing the models. What all you can observe in this model? Any one of you? Any idea? Yes. Hello. What all structures can you see here? Okay. Go back to your general embryology lectures. Just think about them, what all you studied there, and then we'll revise. Like, there is, before fertilization, there are certain things. And then after fertilization, what happens? Zygote is formed. So, this is the male and female pronucleus along with the two polar bodies. Okay, the green colored structures are the polar bodies, and these two are the male and female pronuclei, plural is nuclei. And they are enclosed by this blue colored thing. What is that? It is zona pellucida.
Zona pellucida.
Very good. Now, what is the function of zona pellucida?
It prevents it prevents ectopic pregnancy.
Very good.
Pregnancy.
Yes, and it prevents. This, see, trophoblast has this tendency to eat up everything that is surrounding it. So to prevent that erosion of the trophoblast, the zona pellucida is there. So as soon as the zona pellucida, it hatches out from the zona pellucida, it gets implanted in the uterine endometrium. So it prevents abnormal implantation, that is ectopic. Okay, and it also prevents. What is the second function of zona pellucida? Anything? Speak up. अगर आप बोलेंगे नहीं तो आपको confusion कभी clear नहीं होगा.
मैम
कुछ भी
क्लीवेज
कर,
में
क्लीवेज
मैम
बाद में आता है, क्लीवेज का भी नहीं मतलब है। उससे पहले क्या है, जोना पेलिस्टा के दो रोल्स हैं। इट प्रिवेंट्स पॉलीस्पर्मी।
Yes, from ZP3 is suppressed.
Yes. Now, this is another one. So this one. This is what happens is cleavage is there, and 2 to 4 to 8 to 16 cell stage is reached. The 16 cell stage resembles mulberry. So it has been given the name of morula. Okay, so this is morula, 16 cell stage surrounded by zona pellucida, and this is morula. Now this morula arranges itself into an outer layer that is known as outer cell mass, and the inner one is known as inner cell mass. Okay, that is why it is shown in two different colors. Fine. Now this inner cell mass arranges itself towards a pole. It becomes eccentric and arranges itself towards a pole. That pole has been given the name of animal pole or the embryonic pole, because from this inner cell mass the embryo develops. So this is animal pole to which this inner cell mass is attached, or the embryonic pole. The one opposite to it is known as abembryonic pole or the vegetative pole. Clear? This is surrounded by the trophoblast, that is the outer cell layer or the outer cell mass that converts into trophoblast, right? This is the similar thing, but additional structure is found here that is known as blastocyst. So this stage is known as blastocyst, where you have a cavity that is because of when the implantation has taken place, the uterine and fluids, they imbibe inside and they push the animal pole towards one side, creating a fluid filled space that is known as blastocyst, and the stage is known as blastocyst. Now this is the trophoblast with the inner layer and the outer layer. So how many layers are there in the trophoblast?
मैम 2 लेयर्स होती हैं
2 layers होती हैं, क्या क्या होती है?
मैम स्फिंक्टर ट्रोफोब्लास्ट एंड साइटोट्रोफोब्लास्ट
तो अंदर कौन सी होती है?
मैम साइटोट्रोफोब्लास्ट
Cytotrophoblast is inner, and the
सिंचियोट्रोफोब्लास्ट आउटर
syncytiotrophoblast is the outer layer. Okay. Syncytiotrophoblast is the one that is multi-layered, and cyto is single-layered. It keeps on adding cells to the syncytiotrophoblast. That is why it resembles the syncytium. Syncytium में क्या होता है? सेल का लेवल तो एक ही होता है, लेकिन वो क्या होते हैं? न्यूक्लियाई आर प्लेस्ड एट डिफरेंट लेवल्स. दैट इज व्हाई दे गिव द अपीयरेंस ऑफ मल्टीपल लेयर, बट इट इज नॉट एग्जैक्टली द सेम. ठीक है? This is a similar one. Another model over here again. This one. See the blastocyst, and here this is the inner cell mass. Again has formed a cavity. That cavity has been named as amniotic cavity. And when later stage is coming, this blastocyst turns itself into primary yolk sac. Okay? Up till now, the embryo, this embryonic disc is not properly formed. This is second week of development, where there is epiblast, the blue one, and the hypoblast, that is red one. So hypoblast is contributing to the primary yolk sac, forming a secondary layer that is the Hughes membrane. This Hughes membrane, when it is formed, is responsible for the formation of secondary yolk sac. Whereas this amniotic cavity is the one smaller in shape, size, and the yolk sac is larger in size. Amniotic cavity is towards the epiblast and the hypoblast is for the yolk sac. Right? And this is the cyto and syncytial trophoblast. Now here the cells are shown. Amniotic cavity के side में epiblast के cells कैसे होते हैं?
Columnar.
Columnar. So this one is columnar. That means this is the side of amniotic cavity connected to the yolk sac. The connecting stalk. And the one below is, that is hypoblast. These cells are of
Cuboidal.
cuboidal. That is how you can differentiate between the two. Now this uses membrane demarcates and forms the secondary yolk sac, so it becomes shrunken in size and reduced in size. Okay, and then there is this cavity is formed that is known as extraembryonic coelom. Initially, it is extraembryonic mesoderm, but because of the cell death, the small cells they die and they form small cavities. As in the case of seed, you see the small holes are there, and if you police all those holes, they become a single large space or a cavity. Similar is the case with extraembryonic mesoderm. Cell deaths occur over here in whole of the mesoderm, and they produce small cavities. Now these small cavities, they coalesce together, they come together and form a larger cavity that is known as extraembryonic coelom. Right? This extraembryonic coelom is divided into planktopleuric extraembryonic and somatopleuric extraembryonic mesoderm. So this is a third week of development where the mesoderm has formed. This is a bit advanced stage of third week where you can see the neural groove, notochord. The one having the neural groove is ectoderm. This is fish tail shaped mesoderm, and this one is yolk sac. Okay. A similar thing is shown over here. Cuboidal and columnar cells, amniotic cavity and yolk sac. It is surrounded by Hughes membrane. Again, this is the thin connecting stalk. The smaller one is amniotic cavity and this one is yolk sac. Now, how you differentiate yolk sac from the amniotic cavity? Can anyone explain? बोलिए कुछ एक्स्ट्रा दिख रहा है यहाँ पर. This is allantois. See the one a diverticulum from the yolk sac going into the connecting stalk. This is allantoic diverticulum. So this is present only in yolk sac. It is not there in the amniotic cavity. So how you differentiate? Because of this allantoic diverticulum. Now these red arrangements, these are chorionic villi. Chorionic villi, how many types of chorionic villi are there? I mean, on the basis of cyto, syncytio and extraembryonic mesoderm. How many types of villi, chorionic villi are there?
Ma'am, primary, secondary, and tertiary.
How do you differentiate? See this cyto and syncytio trophoblast, they are like finger-like projections. They are invaginations of cyto and syncytio. Okay. As in this case, you see, this is the inner one and the outer one. This appearance is like this only. So this outer syncytio is surrounding the cytotrophoblast. When this is the case, then it is primary villus. Now, when this primary villus cytotrophoblast is invaginated by the extraembryonic mesoderm also, then it becomes secondary villus. And when the blood vessels also penetrate this tuft, then it becomes tertiary villus. Okay? Clear? Is it clear to everyone?
Yes.
Okay, now this is another model showing the same thing. Same chorionic villi is there. Similar one. So this is embryonic disc, third week of intrauterine life. Embryonic disc at first is round. That embryonic disc comprises of epiblast and hypoblast. ठीक है? ये जो आपको ये दिखाया, this is the cut section from the top. That is, the amniotic cavity has been removed and the yolk sac is removed from below. So this is just like a disc having epiblast and the hypoblast. और ये ऊपर यहाँ जो है ये पूरा amniotic cavity होती है and below one is the yolk sac. Okay, there. This is the connecting stalk, suppose. So this is epiblast and this one is hypoblast. Fine. Now this together is known as embryonic disc. It is round in shape initially, and then it becomes oval because of the migration of cells from the caudal end to the cranial end. ठीक है? तो embryonic development की जब बात करते हैं, तो it becomes cephalic and caudal or cranial and caudal instead of anterior, posterior, superior, inferior. ठीक है? ये नहीं होता है embryo में. Embryo has got cephalic and caudal, मतलब head end or tail end, right? So this is the procaudal plate, and this one is primitive streak. The primitive streak, you can see the arrows towards the cephalic end. So these cells are migrating towards the cranial end or cephalic end, and it helps in giving the shape to the embryonic disc. It becomes pear shaped. ठीक है? Pear देखा है? What is pear? Pear shaped is बताइए भाई. It's a fruit. अब आप ही बताइए तभी आगे बढ़ूंगी मैं. What's pear?
Fruits.
I told you so, but which fruit? हिंदी में क्या बोलते हैं उसे?
नाशपाती.
कैसी होती है? एक तरफ बल्ज होता है और दूसरी तरफ tapering end होता है. This is how the embryonic disc looks like. See, it becomes like this. So this is amniotic cavity. Here the cranial end or the cephalic end. This is the procaudal plate, and this one is the cloacal membrane. ठीक है? This is developing brain and the spinal cord. Primitive streak having the migration of cells indicated by an arrow towards the procaudal plate. Again, this is the embryonic disc only with the procaudal plate, cloacal membrane, primitive streak, and the primitive knot, and this is notochord. And this one is showing something different. What could it be? Procaudal plate or cloacal membrane, primitive streak, primitive knot, notochord, developing brain. इतना तो है. But what these three colors depict? कोई कुछ सोचिए, कुछ बताइए. Okay, you type in the chat box. क्या है ये? बताएंगे नहीं तो फिर मुझे कैसे पता लगेगा कि आप समझ रहे हैं? हम्म? इतना जो अब तक बताया वो समझ में आया कि नहीं आया?
मैम लास्ट वाला मॉडल समझ में नहीं आया था।
लास्ट वाला मॉडल समझ में तो पूछ क्यों नहीं रही है? पूछेंगी तभी तो मुझे पता लगेगा ना कि आपको समझ में आया कि नहीं आया। एंड यू कैन ऑलवेज आस्क, इट्स नॉट अ बिग डील। आपको पढ़ना है, समझना है तो आपको पूछना भी होगा ना। आप ही तो पूछेंगे तभी तो मुझे पता लगेगा कि आपको आ रहा है समझ में कि नहीं आ रहा है। Okay, I'll repeat that model, but actually this model is from a later stage, तो आपको थोड़ा सा डिफिकल्टी इसलिए लग रही है। अगर आप जनरल एम्ब्रियोलॉजी को रिकॉल करिए तो समझ में आ जाएगा। Still I explain this way to you. Previous one, this one,
यस मैम।
एम्ब्रियोनिक डिस्क पहले राउंड होती है, उसके बाद वो पियर शेप हो जाती है। क्यों हो जाती है पियर शेप? जो सेल्स कॉर्डल एंड पर होते हैं, प्रिमिटिव स्ट्रीक बेसिकली एपिब्लास्ट में अपीयर होती है कॉर्डली, ठीक है। इट इज सम स्पेशलाइज्ड सेल्स, दे बिकम converted to primitive streak and they allow migration of cells. इसी primitive streak से कुछ cells, ये epiblast और hypoblast है, इसके बीच में insinuate कर जाते हैं, giving rise to mesoderm. So this epiblast becomes ectoderm. It gives rise to mesoderm from the primitive streak, and then the lower one, that is hypoblast, converts to the cells from the primitive streak are replacing the hypoblast and they form endoderm. So basically epiblast से ही सारी चीजें derive होती है, ठीक है। epiblast convert होती है ecto में, primitive streak से mesoderm बनता है। जो यहाँ पे क्या होता है, जब आप छोटे बच्चे होंगे, आप पेरेंट्स अगर सो रहे हैं या आपके बीच में दो बहन भाई हैं, तो आप अगर बीच में उनके घुस जाएं तो क्या होता है, इंसिनेशन होता है ना। बीच में कोई लेटा हुआ है, आप सडनली जाके बीच में इंटरप्ट कर दें, तो व्हाट इज दैट? This is the same thing that happens when the epiblast and hypoblast has to convert to three-layered structure, that is gastrulation is taking place. तो gastrulation कैसे होगा? तीन लेयर्स बननी है ना, तो epiblast में ही कुछ कन्वर्जन होगा ना। epiblast में टेल एंड पे primitive streak अपीयर होती है। primitive streak जब अपीयर होती है 14th डे पे, सेकंड वीक, एट द एंड ऑफ सेकंड वीक, दैट इज स्टार्टिंग ऑफ द थर्ड वीक, to begin gastrulation. Gastrulation is the process of formation of 3 germ layers, ठीक है। तो जब ये यहाँ से primitive streak से cells कुछ नीचे आए ऐसे, और यहाँ इसके बीच में आके insinuate करता है like this. This one is mesoderm. फिर कुछ और सेल्स हाइपोब्लास्ट को रिप्लेस करके उसे एंडोडर्म बना दिया, तो वो 3 लेयर स्ट्रक्चर हो गया। अब वो 3 लेयर स्ट्रक्चर हो गया, ये तो यहाँ रिप्लेसमेंट हुआ। कुछ सेल्स जो हैं वो यहाँ, ये टेल एंड है और ये हेड एंड है, दैट इज केफालिक एंड। तो इसमें यहाँ से सेल्स का माइग्रेशन शुरू हुआ प्रिमिटिव स्ट्रीक से, और ये माइग्रेट होकर यहाँ आ गए। तो वो ये शेप जो है, जो राउंड था, वो एग्जैक्टली पियर शेप हो गया, ठीक है। अब ये जो प्रोकॉर्डल प्लेट है, प्रोकॉर्डल प्लेट इज सेपरेटेड फ्रॉम द आउटसाइड बाय द स्टोमोडियम और द बकोफेरेंजियल मेम्ब्रेन, ठीक है। और यहाँ क्लोइकल मेम्ब्रेन से होती है, दिस इज क्लोइका। क्लोइकल मेम्ब्रेन से आउटसाइड से सेपरेशन होता है। जब ये दोनों मेम्ब्रेन्स रिजॉल्व हो जाती है तो एंटीरियरली माउथ की ओपनिंग एक्सटीरियरली हो जाती है और टेल एंड पर एनल ओपनिंग हो जाती है। This point, that is procaudal plate, along with the cloaca and the notochord, these three structures are the only structures which are devoid of mesoderm. इनमें mesoderm नहीं होती है, मतलब ये जो है एक्टोडर्म एंडोडर्म है, यहाँ भी एक्टोडर्म एंडोडर्म और नोटोकॉर्ड जब बन रहा था वहां भी एक्टोडर्म एंडोडर्म होती है, mesoderm नहीं आती है। ठीक है, these are three exceptions. ये नोटोकॉर्ड है, ये क्लोइकल मेम्ब्रेन है, ये प्रोकॉर्डल प्लेट है। ठीक है, is that clear now?
Yes, ma'am.
और किसी को कोई कंफ्यूजन? विल स्टॉप हियर, हम इतना ही देखेंगे तभी हमें समझ में आएगा। अगर हम सारे मॉडल्स एक साथ देखेंगे वी कैन नॉट रिकॉल एवरीथिंग, सो विल गो टू द वीडियोस। ठीक है, ठीक है, समझ में आ गया सबको?
Yes, ma'am.
नो रिस्पॉन्स फ्रॉम रेस्ट ऑफ द मेंबर्स। ओके सना, बढ़िया। Okay, so then I'm going to show you a video. टूथ डेवलपमेंट की वीडियोस आपने देख ली? गूगल क्लासरूम से किसी ने देखे या अपना यूट्यूब से खुद सर्च कर लीजिए। देयर इज अ नंबर ऑफ वीडियोस टू मेक इट इजियर फॉर यू टू अंडरस्टैंड। I've shared those videos in the Google Classroom also. यहाँ not able to find. Try to say. So see this video. I'm sharing the feedback form here. You please fill it just now. I've shared it in the chat box. So that's it. Attend your roll calls, please. Mark, some of you, as I've seen. Mariam is there, Nabila, Faiza is there. Who else? Someone is there? Saman Afaq?
Yes, ma'am.
Saman Altaf? No? Prerna? Anas is there.
Yes, ma'am.
Then who else?
I'm Zainab Rauf.
Zainab, what's your role? Or Aditi is there. Rutik?
Anna Nasreen.
Annam Nasreen. What's your roll number? What's your roll number? Sana Nasreen. Huh?
Ma'am, 35.
Huh, I've marked. Rutik was there or not? Rutik Sharma. Saman Afaq, Saman Altaf, Sana Nasreen. Saman Afaq, Saman Altaf, Sana Nasreen. Zainab Rauf, Mohammed Nadeem. Mohammed Nadeem.
Yes, ma'am.
Nadeem, what's
Yes.
your roll number?
I'm 034.
Aditi Vaishnav, roll number 38.
38 marked. नदीम, आपका रोल नंबर क्या है?
मैम 034 2K19 बैच.
ये हुजैफा नदीम?
मैम मोहम्मद नदीम.
तो यहाँ तो नाम है नहीं आपका, मुझे लग रहा है। देखिए मैं स्क्रीन शेयर कर रही हूँ, आप देखिए अपना
ओके मैम.
नाम देखिए जरा। See, 2K19 has got Fuzail Ghani, Harish Malik, Huzaifa Nadeem, Nusrat Ali and Anshika Singh. आपका नाम तो है नहीं, रोल नंबर क्या है आपका?
मैम 034 है 19 बैच में.
ये एडमिटेड बैच 2K19 034।
यस मैम.
ये मुझे बताइए फैकल्टी नंबर। आपने अभी तक कोई क्लास नहीं करी?
मैम की ये क्लास
तो कहाँ पे अटेंडेंस लगी है आपकी?
19 FMBD GL 2211.
GL मोहम्मद नदीम।
जी मैम.
इससे पहले आपने कौन सी क्लास करी है? क्योंकि 19 में तो कोई ख्याल से फर्स्ट क्लास के बाद बच्चे आए ही नहीं है। और आपने पहले क्यों नहीं बताया कि आपका रोल नंबर ये है? बट मार्क्ड अगर आप आए थे तो। और कोई बच्चा है? आप लोगों ने फीडबैक फॉर्म भर दिया? रिप्लाई प्लीज। सिर्फ ज़ैनब को
Yes,
बुलाया।
ma'am. Yes, ma'am.
Okay. So we'll continue with this discussion of general embryology models in the next class, inshallah. Okay. Don't forget to fill the feedback form. Thank you.